how cilia coordinate. So they coordinate into metachronal waves. So I'll give a bit of an introduction as to what that means and show you some mathematical tools that allow a connection between the properties of an individual cilium and the emergent wave. So to start with an introduction, I guess quite a lot of you will know about cilia already, but they're active organelles that you find on the surface of things like microorganisms. They're in epithelial tissue, so you find them in your lungs, in various parts of the body. Adam's talk earlier, in one of the stages of his parasite, showed an image with lots of cilia on his parasite. So you get them all over the place, and they beat they look like a filament, and within the filament you get molecular motors. So they beat by bending the filament, but those motors are within each individual filament. So each individual filament is beating via motors within that one filament. So the question is, how does that filament, which has motors inside it, coordinate in a coordinated manner with the other cilia in that same part of the body or on that same microorganism as it? And what we see is that these things beat in this metachronal pattern. So what that means is, if you characterize the beat of an individual cilium with a phase, so the phase tells you how far through the beat cycle you are, then a metachronal wave means a traveling wave of the cilium phase. So you get a wave that moves along the cilia surface. So we're not talking about a wave in the fluid, we're talking about a wave in where the cilia are through their beat cycle. And in this image here, the white arrows show the peak in cilia tip height. So what you see is, as the cilia goes through its cycle, there's some point in time where it will find the peak height. That's where the arrow is. And as time evolves, you can see that the cilia with the highest tip is moving along that body. So that's the metachronal wave. And here's just an image that shows you that you can find cilia all over the human body. They do lots of important tasks in biology. So for humans, well, other animals as well, but specifically, if you want to think why they're important in humans, they're important for symmetry breaking in embryonic development. They move signaling molecules around the brain. They transport over through the fallopian tubes. They remove things like mucus from your lungs. So these are just, as you can see, they're really important. And they do lots of different functions, biological functions. And as well as in animals, they're also in lots of aquatic organisms, but on the outside, not on the inside, with your small aquatic organisms. As you can imagine, they are doing all these important functions. So if they go wrong, that can cause disease. One thing about the metachronal coordination is this metachronal coordination is particularly important for efficient fluid flow. So the purpose of these cilia is to create fluid flow for various tasks that I've just mentioned. But if you want efficient fluid flow, then there's various groups who in the past have shown that the metachronal coordination is the way that you can get efficient fluid flow. So if you want efficient fluid flow, you need that coordination. So if we want to know how things work, if we want to be able to understand how or why things go wrong, it's important to understand how these things work properly. So the goal of my work is to understand how the properties of an individual cilia beating affect the collective behavior. So there's two questions, which is, one is, how do cilia coordinate to produce this metachronal coordination? And can we tune the properties of an individual cilium beat to get some desired wave outcome? The second question is, I guess, more in terms of if you're using microfluidics and you want to use artificial cilia to create some kind of flow, that's the kind of thing you might be interested in. But in terms of natural cilia, we're interested in how these things emerge. So there's two main mechanisms that these cilia can coordinate. One is hydrodynamic interactions, and the other is mechanical interactions through the substrate. I'm not going to talk about mechanical interactions. I believe Luke, who's giving the next talk, will tell us about that next, which is great. And if you want to know some more about that story, then talk to Kirsty Wan as well, because she's done some lovely experiments on that side of things. But I'm going to talk about hydrodynamic interactions today. So the model I'm going to use is this rotor model of cilia. So what you can see on the left is a schematic of these filaments that are beating. And I'm going to map that to a problem where you have a spherical bead moving around a circular trajectory. And this is something that is used quite commonly in theoretical modeling for cilia, and it's very effective in capturing quite a lot of the important dynamics. And the nice thing about this is we can write down, it's much easier to write down the hydrodynamic interactions when we have this model. So this is the setup I'm going to use. So we've got these circular trajectories, and we've got these beads that are moving around the circular trajectories. And we can use some geometrical parameters to describe these trajectories. The most important one that I'm showing you here is phi. So phi, the variable phi, is telling you how far this bead is moving around this trajectory. So that's telling you, if you map back to the cilia problem, is how far you are through the beat cycle. So phi is how far you are through the beat cycle. So we're looking for a metachronal wave in phi. So that's the important parameter. Theta and chi, they're just how you orient these trajectories on your lattice. So you put all these cilia on a lattice. Theta tells you the direction it's pointing in. Chi gives you some tilt. But phi is the thing that we're looking for a wave in. Now we can write down how the phase of each cilium evolves in time. So if we just choose a cilium on the lattice, cilium I, the time evolution of the phase has some intrinsic velocity, and that depends on this intrinsic driving force that's moving it around the trajectory. That's what the molecular motors are doing. And then the zeta is the friction, so the hydrodynamic friction this bead is feeling. And A is the size of the trajectory. So if you've got a large amplitude oscillation, you've got a big A. If you've got a small amplitude oscillation, that's a small a. The second term is hydrodynamic coupling between all pairs of rotors. This kernel G is the Blake tensor. The reason we use the Blake tensor is because you find cilia on surfaces. So the Blake tensor accounts for hydrodynamic coupling between 2-point forces above a substrate. The beat pattern and the friction, they are both periodic in the phase phi. So we can write them both as a Fourier series. In the stuff I'm going to show you later on, you're going to see these ANs and BNs and CNs and DNs appearing. So if you see them, the thing to remember is that they are the amplitudes of the harmonics in the force pattern and the friction. Okay, so we can write down that dynamical equation. But how do we go from this dynamical equation to saying something about the emergent wave? So I'm going to take you through some steps next, which are the mathematical steps that I go through to get from what we've got here to telling us something about the wave. So what I'm looking for is I'm looking for a dispersion relation for the wave, and I'm looking for a stability analysis. So I'm going to do a linear stability analysis. So if you get lost somewhere in the technical detail, if you're a biologist and it's unfamiliar, then that's what I'm aiming for. So I'm aiming to get from this is how an individual cilium behaves to how does the emergent wave behave. So that's where I'm going. The first thing is if we're looking for an emergent wave, we want it to have some, that wave is going to have some frequency associated with it. And the first thing to notice is that this equation for phi has an intrinsic velocity that is not constant. It depends on the phase. But what we can do is we can choose a new coordinate which has a constant intrinsic velocity. So basically, I'm just choosing a new phi. So if I'm mapping this beat cycle to a rotor moving around a circle, I'm not restricted to having to choose one particular phi. So I'm just having a new phi to capture where I am in the cycle. And this just makes the mathematics easier. So I've got phi bar as my new coordinate. Phi without a bar is the old coordinate. And so I can write down this relation between them and integrate it up to get this equation there. So now just, I said that phi is the important variable. I'm actually thinking about phi bar now, but phi bar is just some way of characterizing the phase, so where you are in the cycle of the cilium's beat pattern. Beat, like where you are in the beat cycle. So then you can do some more mathematical steps to write down this dynamical equation for an individual cilium in this new coordinate. I'm going to go into 2D Fourier space, which means that you're going to start seeing these little Qs appearing. So yes, if you see Qs, that's where they come from. This step here is an important step in the analysis. So what I'm thinking about is I'm thinking about the long-term dynamics. So if I'm thinking about, you've got all these cilia going around doing their thing, but I'm looking for a wave that emerges, and I want to see what happens after that wave's emerged. Where does that, what wave has emerged out of this system? And that happens on a longer time scale. So I'm going to do a separation of time scales between the mean phase of each pair of rotors and the phase difference between pairs of rotors. And the phase difference evolves much more slowly than the average phase. So we can average over the mean phases. And yeah, so if you do look at simulations, the waves emerged over hundreds to thousands of individual beat cycles. So if you're worried about that step, that's why that step is justified. Just another change of notation. If instead of using I to index each cilium, I'm going to use its position on the lattice. So little r is the position on the lattice. So it's just a way of keeping track of all the different cilia. So doing these mathematical steps, I've now got a new equation, a dynamical equation. So I haven't really, I haven't got anywhere towards the wave yet. I've still got an equation for a single cilium here. But now I'm only having to worry about phase differences. I'm not having to worry about individual phases. And what we can see here is that you can put all the geometric information into this prefactor M and this prefactor S. And we can look at the forms of these. They're really horrible, messy expressions. But if you if you look at them, then what we actually find is that these coefficients, these G0s, G1s, G2s, they contain just the direction that each cilium parameter is facing. But the forcing parameters, so that's what went into those Fourier expansions I showed you. The only one that comes up is the second harmonic. So all the other harmonics that were in the force pattern, they got lost during the time averaging process. They all cancelled to zero. So the only thing that's coming out on long time scales is the second harmonic. And this has important symmetry implications that I will talk about in a few slides. And the other thing on that symmetry point of view is what I'm doing here is I'm only doing things to leading order in the trajectory size. And that's feeds into the symmetry of that second harmonic there. I'm just going to skip over that slide for now. But I can come back to that in the questions if you're interested. So what I'm going to do to try and get to a dispersion relation is now I'm going to put in this traveling wave ansatz. So this here is the form of a traveling wave, omega t minus k dot r, and then I'm going to put a perturbation around it. If I insert this into my dynamical equation and then expand to 0th order in the perturbation, so that means neglect the perturbation, and then I can get a dispersion relation. Again, this second point here is just another mathematical step to make the calculations easier. If you're interested in what this means, I'm just doing the sum over reciprocal lattice space instead of real lattice space. If you don't know what that means, don't worry. The sum over G means I am counting all the different pairs of interactions. It's just a different way of calculating that. So anyway, we get this dispersion relation here, so that we've got some way of calculating a dispersion relation now. And if we look at the shape of that, this is what we get here. So we get higher frequency at small wave numbers. So small wave numbers are large wavelengths, and that means that neighboring cilia have they're very close together in their phases. So if they're pushing each other, their flows that they create helps push their neighbors around. If you get large wave vectors, that means you have small wavelengths. So neighboring cilia have bigger phase differences, so they don't help each other go around as quickly. In order to look at the stability, we expand dynamical equations. So we put that wave ansatz back in, but now we go to first order in this perturbation. And then we can get this out, this equation here. So this equation, this is just, this here is the Fourier transform of, it's the Fourier transform of the perturbation. But we've got a time evolution equation for this, and we've got this prefactor, which we can look at that and ask whether it's negative or positive to say whether the perturbation grows or shrinks. So for a given wave vector k, if we want to know if a wave vector k is stable, then we need this prefactor to be negative for all perturbation wave vectors Q prime. So if this thing is negative for all Q prime, then the wave vector K is a stable metachronal wave. If there's any wave vector Q prime that makes this coefficient positive, then that perturbation will grow, and that means that the wave vector K is not linearly stable. So that's our stability condition, and we can plug that in, and that's what the colors represent. So I didn't explain the colors before. So here's some just plots with different examples. The arrow shows the direction of the individual beat, and what we see is that the stable blue regions are in the same direction as that beat, or in exactly the opposite direction. And the reason why we get that forward-backward symmetry is because we've only got the second harmonic, and if you have the second harmonic, then you get a phi and a phi plus pi symmetry. So, well, okay, that's the symmetry of the system that we've got. But if we've got, you know, directionality is important with these things. If you've got cilia in your lungs, you want to make sure the mucus is moving in the right direction. So it's important to be able to distinguish between the forwards and the backwards direction here. So what's going on? Well, what's going on is that I've only so far expanded to leading order in A, which basically means that each cilium is actually just a point force rotating round on a point, is not capturing the size of A. So the next thing I'm going to do is I'm going to go to the next order in A so that I can capture the size in A, which means my point force is traveling as it rotates. So that changes the symmetry of the system. And when I do that, I get all these additional terms in. This is just again, this is a dynamical equation for a single cilium at point R. Before I just had what I'm now calling M2 and S2, but now at order A, I'm getting M1, S1, and I'm using these subscripts to denote that I'm getting the first harmonic. So I'm getting the second harmonic at leading order, and I'm getting the first harmonic at the next order, at the linear term in A. So when I go to the next order, there's a change in the symmetry of the system, and we're getting the first harmonic in. And when we put the first harmonic in, we break this phi to phi plus pi symmetry, and now we get a direction. So this is just a particular example, and you can see the blue region, that's the stable region, but we can see we only get the stable region in the forward direction. And the shape, this is also showing the shape of the dispersion. So you can see there's a symmetry breaking in the dispersion relation, and there's a symmetry breaking in the stable region. And you can change the parameters to move where that stable region is. So here, compared to the last slide, here I've got my first harmonic coefficients in the forcing as positive. So this is the force pattern has positive, the first harmonic in the force pattern has positive coefficients. And in this example, it's got negative. So you can flip the sign, and that flips the stable region. So yes, to summarize, cilia can coordinate their beat cycles into metachronal waves through hydrodynamic interactions. This is something you can see in simulations as well, and people can do things with much more highly resolved filaments and stuff, but we're seeing this again in the analytical model. But with the mathematical modeling, we can relate the individual cilium properties to emergent wave properties, by which I mean the dispersion relation and the stability analysis. And the forcing pattern, so that Fourier series that I showed you right at the start, that tells you how the force varies as the cilium moves through its cycle. And what we find is that the second harmonic is dominant for the coordination. That's what comes out at a leading order, but actually that doesn't have it doesn't have enough of the symmetry properties we need to be able to distinguish between forwards and backwards. So we need to go to higher order to break that symmetry, and then we can see the directionality to that metachronal wave. Yeah, and I should acknowledge the people I worked with. So these are the people who I worked with on the leading order problem. And yeah, so that the leading order stuff is published. The next order stuff is not published yet, but that's the latest. And that's the funding as well. So thanks to EPSRC and my fellowship. And thank you for your attention. And I'm happy to take questions if there is time.